Welcome to the unit on Mendelian genetics. In this unit, we will discuss the science of inheritance, including the work of Gregor Mendel, who is considered the father of modern genetics. In this chapter, you will learn how to work with genetic information and complete genetic inheritance problems, including monohybrid and dihybrid crosses and human pedigrees. We'll begin by looking at Mendel's work and his principle of segregation. Transmission genetics studies the way genetic differences among individuals are passed from generation to generation. Artificial selection, the selection of favorable characteristics in crops and domestic animals by humans, has existed for many years before we understood how traits were being passed from generation to generation. We can see this in ears of corn, where big ears and soft kernels were selected for over many generations, or in domestic dogs, which have numerous traits selected for, generating the wide variety of offspring we see between breeds. There were many hypotheses over the centuries regarding the method of transmission genetics. Hippocrates proposed that each part of the body in a sexually mature adult produced a substance that collected in the reproductive organs and determined the inherited characteristics of the offspring, while Aristotle proposed that the process of heredity transmits only the potential for producing traits, not the traits themselves. In the 19th century, most people, including Charles Darwin, believed in the blending inheritance, in which the parent traits would be blended together in the offspring. For example, if a black rabbit and a white rabbit mate, they would have gray offspring, a blending of black and white. The problem with this hypothesis was that if true, variation would be lost over time. If this gray rabbit mates with another white rabbit, their offspring would be less gray. This process would continue over generations until no black rabbits were seen anymore. The pattern of loss of variation is not what is observed in most populations, suggesting this hypothesis was incorrect. Gregor Mendel is considered the father of modern genetics because he was able to show that the blending inheritance model was incorrect based on genetic breeding experiments he conducted from 1856 to 1864 using pea plants. These breeding experiments are also called hybridization experiments. Mendel conducted hybridization or interbreeding studies between two different varieties of the pea plant, focusing on 7 contrasting traits. He looked at the color of seeds, the shape of the seeds, the color of the pods, the shape of the pods, the color of the flowers, the position of the flowers, and the plant height. For each trait, he crossed a plant that was true breeding for one variety, such as yellow peas, with a plant that was true breeding for the other, in this case green peas, and examined the offspring to see what they looked like. True breeding means that the appearance of the offspring in each generation are identical to the previous one if the plant is crossed to itself. In pea flowers, both female and male reproductive organs are enclosed together within the flower's petals, which allows the pea to self-fertilize. However, Mendel needed to control which plants were crossed, so he had to prevent self-fertilization. He did this by removing the anthers, the pollen-producing structure, from some flowers. He then fertilized the stigma of these flowers by taking pollen from another plant and transferring it to the female parent. A cloth bag was then tied around the fertilized flowers to make sure that any stray pollen did not land on the stigmas. For each of the 7 contrasting traits, the original true breeding strains are referred to as the parental, or P1 generation. The offspring of these crosses are called the F1 generation. In this cross of P1 yellow and P1 green, all the F1 offspring had yellow seeds. Mendel saw this same pattern for all 7 traits. Only one of the traits was seen in all F1 plants. Mendel explained these findings, concluding that there was something he called a hereditary factor that controlled the version of the trait. We now know that the hereditary factor that results in contrasting traits are different forms of a gene that affect that trait. The different forms of the gene are called alleles. One of Mendel's key discoveries came when he allowed the F1 generation plants to self-fertilize, because these plants did not breed true. In the F2 generation, the trait that had been lost in the F1 always reappeared. Interestingly, no matter which traits were examined, the lost trait always reappeared with the same ratio of 3 to 1. Mendel used this data to develop his principle of segregation. The principle of segregation states that individuals inherit 2 copies of each gene, one from the mother and one from the father. And when individuals form reproductive cells, the 2 copies, or alleles, separate equally into the egg or sperm. The segregation of alleles reflects the separation of maternal and paternal chromosomes in meiosis I, when the homologous chromosomes separate in anaphase I, and each chromosome goes to a different pole, eventually entering separate gametes. Going back to Mendel's P crosses, we know that each of the parental Ps must have had 2 copies of the P color gene. Because the parental generation was true breeding, this means the 2 copies are the same, so the yellow parents had 2 copies of the yellow allele, indicated by an uppercase Y, and the green parents had 2 copies of the green allele, indicated by a lowercase y. Both of these parents have a homozygous genotype because their alleles are the same. The yellow parent will pass a single big Y yellow allele to the F1 generation, and the green parent will pass a single little y green allele. This gives the F1 generation a big Y little y genotype. The F1 generation is heterozygous, since the 2 alleles are different. In a heterozygous organism, the trait that is seen in the phenotype is said to be dominant, while the trait that does not appear is recessive. In this case, all the F1 generation were yellow, so yellow is the dominant trait. Dominant traits are often symbolized with a capital letter allele, while the recessive allele is indicated by lowercase. When the F1 progeny form gametes, the principle of segregation says that the uppercase Y and the lowercase y alleles will separate during meiosis, so that half of the gametes will contain only the yellow allele, and the other half will contain only the green allele. We use a depiction called a Punnett square to show the allele segregation during gamete formation, and then fertilization to form zygotes. Along the top of the Punnett square are the possible gametes from the female flower, which can contain either the yellow allele or the green allele. Along the side are the possible gametes from the F1 male, which could also contain a yellow allele or a green allele. Inside the boxes will then be fertilization. If the male gamete with the capital Y fertilizes the female gamete with the capital Y, then the zygote will have the genotype yellow, yellow. But if that gamete instead fertilized the female gamete with the green allele, then our zygote would have the genotype capital Y, lowercase y. Alternatively, the male gamete with the green allele could do the fertilization. In that case, we would have a chance for a heterozygote zygote or a homozygous recessive zygote. This gives us a genotypic ratio of 1 to 2 to 1, with homozygous dominant to 2 heterozygous to 1 homozygous recessive. Since we already determined that the yellow allele is dominant to the green allele, this means that we can determine the phenotype based on the genotypes. Any organism with a capital Y will be yellow. Our phenotypic ratio will be 3 yellow to 1 green. Each of Mendel's crosses with his 7 traits was a monohybrid cross. Monohybrid crosses are genetic studies which involve looking at the effect of only one gene. Here is an example of a monohybrid cross. You should pause the video, read the problem, and see if you can complete it on your own before watching me solve it. When doing genetic problems, you always want to pull out the key pieces of information from the problem itself. Here we will write out that black will be represented by the dominant allele, while brown will be represented by the recessive allele. We'll then write the cross itself. We are crossing a black lab with a brown lab and fill in the genotypes. The problem tells us that the black lab is heterozygote, so he must have a dominant B and a recessive B, and the brown lab must be homozygous recessive in order to see the brown color. We then set up our Punnett square. Remember, along the top we have the gametes from one dog, so our heterozygous dog could give us a black allele or a brown allele, and along the side we set up our gametes from the other dog. The brown dog could give us a brown allele or a brown allele. In the boxes we then complete our fertilization. Looking at the square, our ratio of genotypes was 2 heterozygous to 2 homozygous recessive. Note that this can be reduced down to a 1-to-1 ratio, so either would be correct. The phenotypic ratio is also 2 to 2, with 2 heterozygotes being black to the 2 homozygous recessive brown dogs. Sometimes you have an organism with a dominant phenotype, but you do not know its genotype. Remember, a dominant phenotype could be caused by a homozygous dominant genotype or a heterozygous individual. In order to determine if that organism is homozygous or heterozygous, you can complete a specific genetic cross called a test cross. In a test cross, you breed the organism with a homozygous recessive individual, and then examine the phenotypes of the offspring. If the organism was heterozygous, the offspring will have a 1-to-1 ratio of dominant to recessive phenotypes. But if the parent was homozygous dominant, the offspring will all show the dominant phenotype.